Hey everyone, it's Mr. Sinti, and today I'd like to talk to you about some of the classic experiments that occurred many years ago, trying to determine the fact that DNA, as you can see in this picture, um, was the recipe, in fact, for polypeptides. We suspected as much, and so we have this notion of that one segment of DNA codes for one protein or one gene, one polypeptide, and this is the conversation that we're going to have. What's interesting here is this is a picture recently. This is a picture taken by an Italian scientist about 60 years after Watson and Crick determined the structure of DNA, and this is some of our best photography here. You can see the double helix of DNA. It's a twisted ladder. Incredible. And so in this conversation that we're about to have now, I want to talk to you a little bit about some basic principles and how DNA codes for proteins. And so let's dive right into that. We know that DNA is a recipe for proteins, and proteins are chains of amino acids which have these interesting folding ability, different conformations, and then those manifest into the physical, or if you will, the phenotype, or a particular trait, like in this case, iris color. And so I want to take a look at how DNA bridges that gap between DNA, protein, and traits. And so this is what we're going to look at in this conversation. And so taking you back historically just for a moment, you might recall this somewhere in your past in biology, that Gregor Mendel did a lot of experimentation with plants. And it seems simple when we look back on it, but it was rather profound. He was quite the geneticist early on. And so, as it turns out, he was growing these, if you remember this, he was growing these pure tall plants that were breeding true. In other words, he isolated them from other pea plants, and they kept, when he was crossing them and self-pollinating, they were always producing tall. Likewise, he had these set of short plants that were dwarf, and they kept reproducing short. And so the classic was, what would happen if you cross the tall and the short together? And if you remember, they produce this hybrid plant, which was all phenotypically tall. And so Mendel just simply recorded it, but what we know today about this is that the reason that the plant is short is that it lacks a series of hormones called gibberellin, or gibberellic acid. And as it turns out, that if you take a short plant and you add this hormone, it will actually cause the shoot to elongate. And so as it turns out, that there's some sort of breakdown, there's some sort of mutation. In other words, something was wrong with the gene for gibberellin. And therefore, when the protein is produced, it doesn't produce enough, and so therefore the plant is short. And so when you take a look at this, when you cross, for example, a short plant with a tall plant, all the plants here are tall, and they're considered to be hybrid or heterozygous, if you remember that. And then if you take these heterozygous plants and you cross those together, you get that classic tall, 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 short, or the 3-to-1 ratio. And as it turns out, that the reason being that this plant, the reason being that this plant is short, let's see if I can go back here. The reason that this plant right here is short is because both copies of the gene are not producing enough hormone, and therefore the plant is short. And in this case, this plant is producing enough, so it's tall. This is producing, both genes are fine, they're producing tall, and this is producing tall. So we now know that proteins are responsible for things like traits, but historically we didn't know that so clearly. And so I want to take you back over 100 years ago, 1910, and this individual, Archibald Girard, was one of the very first scientists to suggest that some sort of defect in the genes were responsible for sort of different diseases that were being noticed by physicians. And that he took it a step further, and he found that a disease, for example, this one, that causes darkening of the urine. So urine is usually a yellow color, but if you have this disease, then it turns kind of a brownish, blackish color. And as it turns out, he was able to track this and it was within certain families. And then he was able to do a simple pedigree like we just saw a second ago, and he was able to determine that that the gene that was responsible was inheritable. And if it was inheritable, perhaps it was what was causing the disease was a deficiency in the protein, sort of similar to what we saw with the dwarf pea plant. And so he was thinking that maybe there was an enzyme wrong in the individual, and that's why they were coming down with this disease. And so he sort of postulated that mutations can arise in genes, and therefore enzymes are affected, and therefore diseases result phenotypically. Well, where this leads us is to the understanding that we understand biochemistry a lot more these days. I know you do. Like for example, if a product D, for example, needs to be produced, and A is a precursor, we can say that A is broken down into B, and B turns into C, and then C turns into D. We've seen these kind of linear pathways where a precursor produces a product, and we've even discussed how they're regulated. If the product D is in high concentration, it sort of feeds back, and it binds allosterically to one of the very first enzymes in the biosynthetic pathway, therefore altering its conformation and therefore slowing down the production of B, C, and then D. So it's negatively inhibited. And that we know that each of these reactions in the pathway are catalyzed by specific enzymes. In this case, if this is X turning into Y, this would be Xase and this would be Yase. And so this is important to know in terms of the experiment that I want to discuss to you about one gene, one polypeptide. And so one more before we get into that particular experiment. Here's another synthetic pathway. If we have some sort of precursor, it's not drawn in, but it's some pathway leading to the production of the amino acid tryptophan. So this is a crucial amino acid. Our body can make it. In other words, we don't have to eat it. We could take in this precursor, and then in this linear pathway, this produces this, and this produces this, et cetera, et cetera. And then, as you can see here, what the diagram is showing is that when this is in high concentration, it feeds back. And so we looked at enzyme inhibition. We will, in a future conversation, talk about gene regulation, and that's an interesting conversation. So I want to take you back historically to this scientist, George Beadle. And interestingly enough, George Beadle in the 1930s was working with Drosophila melanogaster. You might recall Thomas Hunt Morgan was working with the fruit fly as well. And in this case, Beadle was speculating that a mutation, rightly so, was the causal agent of, here's a fruit fly that's the most common phenotype is red eyes. He sort of was thinking that maybe a mutation causes the production of the pigment for red eyes, and therefore the fruit fly has white eyes. Although he was never able to show it, he sort of was thinking along those lines. It was rather difficult to work with fruit flies. And so, as it turns out, George Beadle teamed up with scientist Ed Tatum, and the two of them decided to sort of look at the effects of mutation in proteins. And so what they were going to try to do is pick a model organism. So they decided to work with, I know it seems somewhat random, but it's very easy to work with small organisms. Like for example, it's easy to work with bacteria, it's easy to work with viruses per se, and it's also easy to work with fungi. And so they decided to work with bread mold, and it's actually a mold called Neurospora, and it causes red mold in bread. And so as it turns out, in the 1940s, they were using X-rays. This is not an actual picture of an X-ray, but they're using X-rays you can't see X-rays. This is a blue ultraviolet light here. So as it turns out, they're radiating bread mold, and basically when you radiate with X-rays bread mold, it kills them. But as it turns out, they discovered a few interesting things about how they can also cause mutations in the mold that appears as if the mold is dead, but in fact it doesn't really die, and we'll take a look. So how they're growing bread mold is that you don't actually need bread to grow the mold. You could actually grow it in petri dishes, you can grow it in liquid media, like for example in the flask, and better yet, you could even grow it in test tubes. And this media, this nutrient broth, you can put anything you want in it. You can put agar in there, you can put amino acids, you can put vitamins in there, you can everything that the mold would love to grow in, and basically it will grow. And so if I were to illustrate this, if you were to grow mold, it would basically look like this. And what's interesting is that this liquid media can form a slant, and so what you see here is the mold will grow on the top, right in here, just as if it would grow on the top of an orange or on the top of bread. And so basically they're growing mold and they're exposing it to X-rays. And so let's take a look at this. So George and Ed found that when they mutated, in other words, they were shining X-ray on these mold, that basically you could determine what was wrong with them by sort of adding a particular supplement to their growth medium. Because what's cool about this growth medium is that you can control this. You could have one growth medium with all the vitamins except for vitamin A, and then you can grow all of them. You can grow mold with all the vitamins except for niacin, or all the vitamins except for B6. So you can control what supplement, like a vitamin or any kind of one a day as a supplement, or sometimes milk is supplemented with different vitamins. You could add supplements to this growth media, and therefore you could sort of figure out what's wrong with the mold. And so let's take a look. So as it turns out, they're able to establish a link that we now know as kind of obvious today. We know that the DNA codes for a protein, and the protein manifests into a trait. And we know that some of these pathways are linear, but they were able to establish this, and they were using the mutated Neurospora, in other words, the bread mold. And just want to point out this term. This is a term that you might be familiar with in the past, but I just want to refresh you. The wild type is like the non-mutated mold, and so the wild type is basically the most common phenotype. Now, that has no problems whatsoever. So you can actually grow the wild type on what's called minimal media. In other words, the bare bones. And what I mean by bare bones is you don't even give them like amino acids. You don't even give them something that they really need. What are you going to give them? You're going to give them a little bit of agar, which is sugar. You're going to give them some salts and a little bit of glucose and maybe one vitamin. And from that minimal media, they're able to, if you remember that pathway, they're able to make amino acids from it. Like they're able to make tryptophan. They're able to make other vitamins from this. So they are able to employ their synthetic pathways in order to still survive on or minimal media. And so this is a picture of George here. As you can see, in his mind, he's thinking that there's a series of genes in the mold that must code for these proteins called enzymes that are able to convert, as we now know, they're able to convert these sort of basic materials over here into essential amino acids or into essential vitamins, and therefore they are able to grow on minimal media. This is sort of what's happening in the wild type. And so they have the enzymes to convert simple materials into crucial amino acids and vitamins necessary for growth. So the point is, if you radiate or if you expose this mold to X-rays, what you could do is you could take the gene out. In other words, you would basically, here's a piece of DNA, you would alter the sequence by shining X-ray at it. And if you alter the sequence, it's obviously going to produce a defective enzyme, and without that enzyme, then there's a problem. And it's like it's not going to make an amino acid, it's not going to make a crucial vitamin, and therefore it's not going to grow. But what's really interesting about this, or fascinating if you will, is that just on surface, like if you're growing mold and you shine X-ray on it, and then you try to grow it in the nutrient media and it doesn't grow, I mean, what are you to conclude from this? You're like, I don't know, maybe I killed it, for example. But then if you add, see, the point is, if you add amino acids and then it does grow, then you're like, whoa, I guess I've somehow disabled the mold's ability to produce amino acids. But it's a more complicated question than that. Like, or in other words, say I mutated the mold and it doesn't grow, and you're like, oh, I killed it. But then you add vitamins. So say you have a 1 a day and you add it to the minimal media, and then the mold is able to grow, and you're like, haha, I've knocked out its ability to produce vitamins. But when you start asking more questions, you're like, well, what vitamin in particular is it lacking the ability to produce? Like if I give it B, will it live? Will I give it A, will it live? And so it really opens up a lot of questions. So it's a simple mechanism, but they were able to come up with a lot of cool things. So they reasoned that if you mutated some of these genes for example, that produces enzyme A, then it doesn't grow. And so as it turns out, when they add certain supplements, so you start off with every growth experiment with minimal media, that way you can control what you're adding. So if they add vitamin A and they're like, hey, look, it grows. So therefore, we must have mutated a gene that produced an enzyme that was capable of turning the simple substances into vitamin A. And so what's interesting is they were growing a lot of samples. And so as it turns out, the culture, which is a word used for describing the population of mold that they're growing in the test tube. Here's the culture right there. When you see a line like this, it means that it's living mold. And so they're like, here is the culture 299, as it turns out, that is growing in minimal media when we add vitamins. And then here we add amino acids and it doesn't grow. So what does this mean? This means that adding amino acids doesn't do anything. It must not be able to make vitamins. Do you follow? Like over here, it's able to, this establishes the fact that it's growing on complete media. It's like, whoo, good thing we didn't kill the bacteria the mold when we mutated it. And you're like, wow, okay, so let's see if it grows over here on amino acids. No. So what does that mean? That means that it is able to make amino acids, but it's not able to make vitamins, because when you add vitamins, then it's able to grow. So here it was clear that it was not able to make vitamins, and therefore it died. And so you're like, the mutation has something to do with vitamins. So this is all that they're looking at. So I find that that's kind of interesting, because these simple experiments were had a lot of thought behind them, even though they were simple in terms of like how to design it. And so let's talk a little bit more. So here's some mold growing on the test tube, and then you're shining some X-rays on it. And so they believed that if they kept doing this, they'd be able to find like all the genes that produced every different vitamin. They would find the genes necessary that would produce amino acids. And so they kept doing this over and over again. And so in particular, they tested each of the cultures. And so here there's no growth, and then here there is growth. And so they needed to do this in order to establish the fact that they didn't kill the mold. We talked about this before. And then this is how they were able to determine which vitamin in particular was the supplement that they needed. So check this out. Here's the mold, and it's alive. So they didn't kill it because it's able to grow on complete medium. But then over here, when you give it amino acids, it dies, which means it's not able to make vitamins. You're like, okay, I can see that. It's not able to make vitamins because you see how there's no vitamins? There's been no vitamins added. So therefore, the mutation screwed up the mold's ability to make vitamins. And you're like, well, let's show that to be conclusive. Let's add vitamins. And you're like, yes. And there they are. They're growing. So now we know that we mutated them and they can't make vitamins. And you're like, well, let's add niacin. And that's a type of vitamin to see. And you're like, well, it still doesn't live. So that must mean that niacin isn't the issue. And let's add riboflavin, let's add thiamine, and then let's add B6. And when you add B6 and it grows, Therefore, you can come to the conclusion that the mutation was screwing up the mold's ability to produce B6 vitamin, because when you give it the B6, it's then able to grow. So these experiments go on and on and on. So to show that this is the case, so this is sort of the background in this. So if you give it an X-ray and it mutates the gene, it's not able to make B6 and therefore it dies in minimal media. But if you add vitamin B6, it's able to grow. Therefore, that's something. And so now, how does this relate to amino acids? I find this to be like the best part of this experiment. Like this, at the end product is the amino acid arginine. You see this down here like that? And so what's interesting is, let me try to illustrate this a little bit. If you took a test tube like this, and again, you go through the whole thing, you have here's the minimal media. Let me give it some color. Nice and yellow. Minimal media. And you're like, okay, let's see if we can mutate the mold. And we're going to mutate the mold and see if it grows. And we grow it on the minimal media, and what do you think? It dies. And you're like, oh, we must have killed it. And then you grow it on complete media, and it lives. And you're like, no, I didn't kill it. And you're like, well, what can it produce? What can it produce? And you're like, well, let's see if we give it arginine, this amino acid, if we give it arginine, in other words, we add that powder into the media, hey, look, the mold grows. And we're like, oh. So we screwed up its ability to produce arginine. And so that's something. But this is even more interesting. What they were able to do was they were able to determine this linear pathway. This is totally fascinating. Let me just walk you through this for a second. So it's this precursor produces this chemical called ornithine, and then citrulline, and then citrulline turns into arginine. And you're like, okay, whatever. Well, check this out. Like, let me see if I can erase this. Yes, I can. That's nice. Okay, so check this out. Say, for example, we were to provide an X-ray. Let's go black for X-ray. We give an X-ray like this, and it wipes out gene A. So the X-ray destroys this gene. So let's think about this. What's going to happen? So without enzyme 1, the precursor is enzyme 1 is all screwed up. So the precursor will not make ornithine. It will not then make citrulline. It will not make arginine. The mold will die on minimal media. And you're like, oh, okay, thank you very much. But check this out. If you were to add ornithine, let's do that. O-R-G. Is that no? I'm sorry. O-R-N. Ornithine. What's going to happen? If you add ornithine, hey, look, then this enzyme, which is fine, turns it to citrulline, then enzyme 3 turns it into arginine. Hey, with arginine, the mold is able to grow. You guessed it. And you're like, yeah, what's the big deal about this? Okay, so check this out further. So do you see where we're going with this? So, okay, same thing. Here's the test tube. Here's the minimal media. Let's take another mold, and this time we'll shoot the X-ray on it like this, and this time it's going to wipe out gene 2. So if it wipes out gene 2, then that enzyme 2 is all screwed up. And so watch this. If you give it the precursor, it'll make ornithine, and then ornithine would not be converted to citrulline, not converted to arginine, and what? The mold lives or dies. It dies. And you're like, wow, I've seen this before. Let's give it ornithine. And you're like, well, I'm sorry. If you give it ornithine, it still dies. And you're like, well, this is kind of a weird mutation. How about if you give it citrulline, though? And you're like, so put a little cit in there. And if I put a little cit in there, then what's going to happen? This enzyme's fine, and so it's going to produce arginine, and like, hey, the mold grows. You're like, well, this is rather complicated. This is what I'm saying. It's a simple design. All it takes is some test tubes, some minimal media, some vitamins, some sugars, and some salts. And it's a lot of thought, though. And so check this out. When you look, if one gene makes one enzyme in this pathway, they were able to deduce. This is incredible. They were able to deduce this linear pathway by determining when the mold lived and died, and based on the supplements that they were providing. And so, just to refresh you, that a mutant can survive if you give it complete growth media. But when you give it minimal media, it doesn't live. And so it's up to you to determine what amino acid is it not being able to produce. And so, like, same thing with the mold. It's able to grow on mold. The mold is able to grow on bread because bread has everything you need. But if in the minimal media, it's a different story. Now, here's another important note. I just wanted to add this. If you're following this conversation completely, and you understood what I was talking about with the pea plants, you'll know that Gregor Mendel was working with diploid pea plants. In other words, they had tall, short. So in other words, they're diploid. They had 2 genes for every protein. So if one gene was mutated, in other words, short, and it wasn't producing gibberellic acid, the plant's still going to be tall because it had enough of the hormone to make it tall. But in this case, what's clever about their experiments with Neurospora mold was that this was a haploid organism. And so they didn't have to worry about dominant or recessive or 2 genes. They were just working with 1 gene, and that's crucial. Because if you knock out the 1 gene, then you know it's not working. Just to point that out. And so here's the punchline, that one type of mutant required just arginine to grow on it. And so they concluded that it was deficient somewhere in the biochemical pathway that normally produces arginine. And so, as it turns out, George and Ed were also working with 2 other individuals. So their colleagues in 1944, Surab, if I'm pronouncing that right, probably not, and Horowitz, identified the pathway and the crucial enzymes necessary to produce arginine. And so they came up, the punchline is, to this 1 gene, 1 enzyme hypothesis. This is a big deal. And so, as you can see here, if you take out that gene, then therefore it's not going to be able to produce ornithine, and therefore no growth. But if you give it ornithine, it's able to grow. And so that's kind of interesting. Mutant 1 dies, but it lives if you give it citrulline, and it lives if you give it arginine. And so you can look at different mutants. And so let me cut right to the chase. This is an image that's a classic, and let me sort of help you walk through it. This part of it is what's in what you're thinking in your mind, and this is actually what you're looking at over here, which is the test tubes. So obviously the wild type is able to grow. If it grows on minimal media, that means it grows if you give it anything. So it's fine. But look at this. Here's a mutant, and you're like, look, I've killed it. It doesn't grow on minimal media. And you're like, no, I didn't kill it. I've given it ornithine. And when you give it ornithine, check a look over here. If you give it ornithine, which is right here, it's able to make arginine. And so it lives. And then check out this one. This is a different mold. This is a different mutant. This one, it doesn't live on minimal media. And I've killed it. And you're like, no. When you give it arginine, it lives. So therefore, you know that something's wrong with the arginine production. And then this time you give it ornithine, and you're like, it's still not growing. And then you give it citrulline, and it lives. And you're like, that's interesting. Let me keep track of that. So if I give it this, citrulline, it is able to make arginine. So therefore, that comes after. This is gene B. I must have mutated gene B. Do you see that? That's incredible. This is amazing that they're able to come up with this. There's 3 genes in the synthetic pathway of arginine. And by looking at these 3 classes of mutants, they were able to tell the sequence of these and what each gene is producing and the enzyme responsible for producing it. And so this research, rightly so, led Beadle and Tatum to the Nobel Prize in 1958 for their discovery of the 1 gene one enzyme hypothesis, which sort of, if you tweak it a little bit, it's one gene, one protein, but that isn't really the way we should look at things. One gene, even though that's the title of the chapter we're studying, one gene, one protein. Because a protein, as you know, could be more than one polypeptide chain. It could be, in other words, quaternary. So there's issue with that. We should really say that it's one gene, one polypeptide. And we call this a hypothesis, but we're pretty confident about it. So I hope you enjoyed that conversation with mold. You never thought mold could have been that interesting. Thanks for watching.